Okay. Sometimes you hear a statistic that just makes you stop and think. Consider this. Scientists have been working on an HIV vaccine for, well, over 30 years now.
That's right, 3 decades.
It's just a huge amount of time and effort. And for so long, finding something that really sticks, that actually works, has been incredibly, incredibly tough.
It really has. I mean, HIV just throws up these unique roadblocks for vaccine developers. Its biggest trick is that ability to mutate so rapidly, to dodge the immune system. It's been a really persistent puzzle.
Yeah. But what's encouraging, what we're looking at today, is that researchers aren't, giving up. They're pushing ahead with some really novel, kind of out-of-the-box approaches. And they're making some significant steps that are actually starting to show some real promise. So in this deep dive, we're going to unpack these new strategies. We're drawing on some key recent scientific reports, particularly findings published in Science around HIV Vaccine Awareness Day this year.
Yeah, our goal is really to get under the hood of these new methods. How are they fundamentally different from what's been tried before? What are these latest early stage studies actually telling us? And maybe most importantly, what could this all mean, even now, for the future of HIV prevention and maybe even treatment?
Because, let's be honest, the standard vaccine playbook, the things that worked for other viruses, they just haven't cut it for HIV. There's a pretty broad consensus now in the scientific community that we need something, smarter.
Yeah.
More sophisticated, more targeted.
Exactly.
So before we jump into the specifics of the new research, let's just quickly recap the core biological challenge. What exactly are these new vaccines up against?
Okay, so the central problem is this. When someone gets HIV, their immune system does react. It makes antibodies. That's standard. But the issue is that most of these antibodies, they go after parts of the virus that are constantly changing, the highly variable regions. So imagine trying to hit a target that's constantly shifting its shape. These antibodies become pretty useless pretty quickly against the evolving virus.
Right, so the immune system is trying, but its main weapons are basically hitting smoke. The real goal, the sort of holy grail here, are these special antibodies called broadly neutralizing antibodies, or bnAbs for short.
Exactly.
What makes them so special?
Well, unlike the others, bnAbs target parts of HIV that are conserved.
Meaning they don't change much.
Precisely. These are critical components the virus needs to function, so it can't really afford to change them. So a bnAb can recognize and neutralize many, many different strains of HIV because it hits these constant, unchanging targets.
Okay, makes sense. But the catch is?
The catch is that very few people, only a small percentage, naturally develop these powerful bnAbs during an infection. It just doesn't happen often on its own.
Okay.
But, and this is the key insight driving the new research, most people do seem to have the starting materials. They have these rare, immature B cells. B cells are the immune cells that learn to make antibodies, right?
Right.
So people have these specific B cells that could potentially mature into the cells that pump out bnAbs. They just lack the right signals, the right instructions naturally.
Okay. And that's where this new idea comes in. Germline targeting.
Exactly.
So instead of just hoping the immune system figures it out, this is more like giving it a very specific step-by-step training manual. You don't just do one shot. You use a sequence of carefully designed vaccine components. They call them immunogens.
Right.
And each one in the series is meant to wake up and then guide those specific rare B cells, kind of coaching them along the path to becoming mature bnAb producers.
It's really about achieving incredible precision. Finding those needle in a haystack B cells and giving them exactly the right nudge to start that journey.
Okay, that framework helps. It's like guided evolution for your immune system, specifically for this task. Let's get into the actual studies now, the ones showing this in practice. First up, the work from Scripps Research and IAVI, William Sheaf's team.
Yeah, so their approach is centered on engineering a specific immunogen. They created something called EOD-GT8 6-dimer. You can picture it as a tiny nanoparticle scaffold, and it's decorated with 60 copies of an engineered piece of HIV's outer protein, GP120.
Okay.
And this engineered piece is very specifically designed to latch on to only those rare immature B cells that have the potential to become VRC01-like bnAb producers. VRC01 is a well-known, potent type of bnAb.
Got it. And this isn't brand new. They've been building towards this, right?
That's right. There was a trial back in 2022, IAVI-G001, that showed a protein version of this immunogen could successfully activate the right precursor B cells for VRC01.
Okay.
Then last year, they reported a big step forward in preclinical work using mRNA versions. They used an mRNA primer based on that EOD-GT8 6-dimer, and then followed it with a different mRNA booster called Core G28V2 6-dimer. And this sequence successfully guided B-cell maturation and got VRC01-like antibodies produced in mice.
So the animal studies looked good, paving the way for the human trials.
Exactly. Which brings us to the phase I trials we're focusing on. IAVI G002 with 60 people in North America and IAVI G003 with 18 participants in Rwanda and South Africa.
And it's really important they're doing this research in Africa too, right? Given the disproportionate impact of HIV there.
Absolutely crucial. So in IAVI G002, they tested that EOD GT8 60-mer mRNA primer. Some participants got just the primer, others got the primer, followed by that different core G28V2 60-mer mRNA booster. They call this a heterologous boosting strategy, using two different components sequentially.
Why two different ones?
Because the idea is the second shot isn't specifically needed to push the B cells further down that maturation pathway that the first shot started. It's a refinement step.
Okay. And the results from G002?
Pretty striking. So all 17 participants who got both shots, the primer and the booster, developed antibody responses of the VRC01 class. And even more telling, most of these were described as elite responses. That means the antibodies had already started picking up multiple genetic changes, mutations, that are known to be crucial steps towards becoming fully functional broad bnAbs.
Wow, okay. And what about the people who only got the primer?
They generally showed less advanced antibody development, still VRC01 class, but not as mature.
So that difference really highlights that this stepwise guidance, the heterologous boost, is doing what it's supposed to do.
Exactly. And Dr. Sheaf himself apparently commented on how surprisingly good the quality of the response was after just those two steps. He said something like, "We didn't anticipate it would be that favorable."
That really drives home that they're successfully kicking off the process.
It definitely confirms they're priming the right cells and that the second different shot helps push them along. Now, looking at IAVI G003, the trial in Africa, which used 2 doses of just the primer mRNA.
Right, no booster in this one.
Correct. The results there were also really encouraging for those initial steps. Almost everyone, 94%, developed those VRC01 class responses and activated the target immature B cells.
Okay, so the primer works well to get things started.
Yes. And here's something interesting. Even without that specific heterologous booster used in G02, the participants in G003 showed surprisingly high levels of antibody mutation and diversity, similar actually to the group in G002 that did get the booster.
Huh. So the primer alone might be doing more than just activating them.
It seems to suggest the primer itself might be powerful enough to start inducing some of those necessary genetic changes in the B cells. Though the G002 results still suggest that specific booster is needed for the more advanced maturation steps.
Right.
But taken together, these two trials give us really solid human proof of concept. This whole stepwise germline targeting idea can find the right rare B cells and start training them down that bnAb pathway.
Okay, that's really clear progress on that front. Let's shift to the second study mentioned in the reports, this one from the Amsterdam University Medical Center, led by Tom Caniels and Roger Sanders.
Right. They also use a germline targeting strategy, but they employed a slightly different type of immunogen.
Different how?
Theirs was based on a trimer structure called BG505, SOSEP-B 4.1 GT1.1. Trimers are engineered proteins designed to mimic how the HIV envelope protein spike looks on the actual virus surface. The idea is to present the target to the immune system in a more natural shape.
Okay, a different way to present the target. And what did their study find?
Their vaccine also successfully triggered the production of those desired VRC01 class bnAb precursors in most of the people who received it. And what's really exciting here is that a good chunk of the antibodies they generated weren't just precursors, they were structurally very similar to the fully mature VRC01 bnAb.
Structurally similar.
Yes, and importantly, these antibodies were actually able to neutralize wild-type HIV pseudoviruses in lab tests.
Pseudoviruses, those are like safe lab-made versions of the virus for testing.
Exactly. They mimic key parts of real HIV infection without being infectious. So seeing neutralization in the lab, even at this early stage, is a very positive sign.
So they're not just activating the right cells, they're getting antibodies that already show some anti-HIV activity in the lab.
Correct. Dr. Sanders had a quote about it saying something like, "Across the participants, we saw antibody production that tells us we're on the right track. We now know we can target the right cells with atomic precision."
Atomic precision. That's quite a statement.
It captures the feeling, I think. Both of these studies, using slightly different tools but the same fundamental germline targeting idea, really reinforce the message. Researchers seem to have found a way to identify and start guiding the specific B cells needed to kickstart the bnAb production line. It's a powerful validation.
Okay, this is clearly significant progress in figuring out how to get the immune system trained properly for HIV. But, with any new medical advance, especially vaccines, we also have to talk about potential challenges, side effects, and what comes next.
Absolutely. And one thing that did come up unexpectedly in the IAVI trials was side effects. Now, overall, the vaccines were reported as generally well tolerated, but some people did experience skin reactions, mostly mild to moderate hives, usually manageable with antihistamine.
Okay, hives. That doesn't sound too unusual for vaccines sometimes.
Right, but the notable part was that in the IVI G002 trial, about 10% of participants developed chronic hives, meaning they lasted for 6 weeks or longer.
6 weeks or more. Okay, that's definitely something to pay attention to.
It is. And the reporting also mentioned a separate study looking at different experimental mRNA vaccines using another trimer immunogen BG505MD39.3. That study also reported hives in about 7% of participants, and in some cases they were persistent, lasting up to a year.
Wow, a year?
Yeah, and an interesting observation from that separate study was that every single person who got hives had previously received a Moderna COVID-19 vaccine.
Ah. Is there a known reason for that link?
Not yet. The mechanism, either for the chronic hives or any potential link to prior vaccination, isn't clear right now. It's something under active investigation. It's just crucial, as the sources emphasize, to report these findings transparently as part of the ongoing analysis without, jumping to conclusions.
Yeah, that's what phase I trials are for, safety first, finding these things out.
Exactly. And beyond side effects, we absolutely have to keep perspective. These are very early stage results. They show the vaccines can start the desired immune training process, which is huge. But they haven't yet been tested to see if this training actually leads to protection against HIV infection itself, not even in animal models exposed to HIV or SIV yet, let alone in large human trials.
And those large human trials, the ones needed to prove prevention, actually works.
Yeah.
They face their own hurdles now, don't they?
They really do. We now have incredibly effective HIV prevention tools like daily oral pre-PEP, on-demand pre-P.
And even newer long-acting options coming, like that twice-yearly injectable lenacapavir you mentioned is on the horizon.
Right. So when you have such effective prevention already available, it makes designing and running huge placebo-controlled vaccine efficacy trials much more complex, ethically and logistically.
Yeah, the landscape has definitely shifted.
It has. And there's also the practical aspect. Even if these multi-step vaccine regimens prove highly effective in trials, rolling out something complex, requiring multiple specific shots over time, might be challenging in real-world public health settings globally, especially compared to simpler methods like a pill or a long-acting injection.
So lots to consider on the prevention front. But you mentioned treatment earlier. Does this germline targeting approach have potential there too?
Oh, absolutely. This sophisticated strategy isn't just about stopping initial infection. It could be incredibly valuable for developing therapeutic vaccines.
Therapeutic meaning for people already living with HIV.
Exactly. The idea would be to use the same stepwise germline targeting process to train their immune systems to start producing high levels of bnAbs internally.
So instead of preventing infection, it would help the body control the virus it already has.
Precisely. It could potentially become a vital part of a combination strategy aiming for long-term viral control without needing daily pills. What people often call a functional cure or long-term remission.
Wow, that's a really significant potential application, broadening the impact way beyond just prevention.
It really is. It opens up a whole other avenue where this technology could make a difference.
Okay, so let's bring this back to you, our listener. Thinking about everything we've just discussed, the challenges, the new strategies, the early results, what's the single biggest takeaway for you from all this?
For me, I think the most important thing is that after so many years of hitting walls, researchers have genuinely found a new angle. They're not just trying variations on old themes. They're using this incredibly precise, engineered, step-by-step approach to actively teach the immune system to make these very specific, very powerful antibodies that it rarely makes on its own.
Yeah.
That feels like a real shift.
Yeah, it feels like moving from, trying to catch this incredibly slippery virus with a wide net to developing something more like a laser-guided weapon aimed right at its critical, unchangeable weak spots. This germline targeting, it's showing it can start that precise training in people.
And seeing that progress, it naturally leads you to wonder.
Right. Which brings us to our final thought for you to ponder. Given how far we've already come with HIV treatment, making it a manageable chronic condition for many, how might the eventual arrival of a truly effective preventative vaccine fundamentally change the entire global picture of the HIV epidemic? Something to think about.